Hi everyone, welcome to our isolation and characterization of DNA lab week 2. After DNA has been isolated, its identity must be confirmed. The objective of this lab is to identify and confirm the presence of the isolated DNA. Therefore, we will perform the following test: Diphenylamine test, test for deoxyribose, molybdate test, presence of phosphate, Purin based test, presence of adenine and guanine. Now let's talk about the structure of DNA and RNA. DNA and RNA are nucleic acids. Nucleic acids are complex polymers made up of monomer called nucleotides. Each nucleotide is composed of sugar molecules containing 5 carbon atoms, a phosphate group, and molecule containing nitrogen that will be referred to as a nitrogenous base. The nitrogenous bases are divided into two groups according to their size: purine, pyrimidine. The large purines, adenine and guanine, molecules differ from each other in their attached group, as do the three smaller pyrimidine nitrogenous bases: cytosine, uracil and thymine. There is no thymine in RNA and no uracil in DNA. Now, as we know the structure of the nucleic acids, it will be easy to understand and confirm the presence of DNA using different chemical tests. Now let's talk about the procedure. First step, we need to centrifuge the lab DNA from last week at 3000 rpm for 3 minutes. And then we need to decant the ethanol and add 10 ml of citrate buffer into the tube containing lab DNA. Chemical test. DNA can be identified chemically with the diphenylamine test, as DNA contains deoxyribose and RNA contains ribose. For diphenylamine test, we will take 6 samples in 6 different test tubes, which are lab DNA, commercial DNA, ribose, deoxyribose, commercial RNA and distilled water. After then, we will add 1 ml of diphenylamine solution in each tube separately and boil for 10 minutes. After 10 minutes, we will mix the solution and observe the color changes. The presence of deoxyribose will give you the brown color. Here, the reaction depends on the conversion of the deoxyribose to aldehyde, which then reacts with the diphenylamine to give brown color complex. Diphenylamine does not react with the ribose sugar in RNA and does not form brown color complex. Next test is the molybdate test, which is done to confirm the presence of phosphate group. For this test, we will take 6 samples in 6 different test tubes, which are lab DNA, commercial DNA, ribose, sodium phosphate, commercial RNA and distilled water. Here, the sodium phosphate is the positive control and distilled water is the negative control. For this test, we will take 1 ml of nitric acid followed by 1 ml of ammonium molybdate solution, and we will boil this sample for 2 minutes. After 2 minutes, allow to cool and then add 1 ml of ascorbic acid solution to each of the test tubes and observe the color changes. The presence of phosphate group is indicated by the formation of blue color. The principle behind the molybdate test is when a solution containing phosphate ions is heated with a solution of molybdate and nitric acid, phosphate react with ammonium molybdate to form a ammonium phosphomolybdate compound and a yellow color precipitate formed. This ammonium phosphomolybdate is further reduced by the ascorbic acid to form a blue color complex. Now we will do the Purin base test. For this test, we need to hydrolyze our nucleic acid samples. This can be done by adding 1 ml of lab DNA, commercial RNA, commercial DNA into 3 separate test tubes. After that, we will add 4 ml of sulfuric acid and boil for 15 minutes. After 15 minutes, allow it to cool before using these samples for the Purin test. For the Purin based test, we will use hydrolyzed lab DNA, hydrolyzed commercial RNA, hydrolyzed commercial DNA, ribose, adenine, distilled water. Adenine is the positive control and distilled water is the negative control. After that, we will add 1 ml of concentrated ammonia solution, 1 ml of 2 molar silver nitrate into each test tube separately. We will mix it thoroughly, then wait for 10-15 minutes to observe the white precipitate formation. The presence of the Purin bases, adenine and guanine, will indicate by the white precipitate. As you know, hydrolysis is a chemical process in which a certain molecule is split into 2 parts by the addition of a molecule of water. Hydrolysis breaks down the nitrogen glycosidic bonds between Purin bases of ribose and deoxyribose. As a result, it releases Purin bases adenine and guanine. After adding concentrated ammonia and silver nitrate solution in a white precipitate form, it indicates presence of Purin bases. Thank you for watching this video and see you in the lab.